Welcome everyone, my name is Matthew Tay from the TU Munich, and the title of my talk is Focus on the Spectra That Matter by Clustering. So I'm happy to announce that this work has recently been published in Nature Communications, so please go there for all the details. So one of the central questions I asked during my PhD was how can we extract more information out of the data we acquire from our mass spectrometers? So the further I got in my PhD, the more I realized the extent to which we are actually throwing away a lot of data very early on in the process. So to tackle this, we came up with a method we call Condenser, where we apply clustering to our raw data to condense our data into a set of summarized quantifications together with spectra of interest. So on the right-hand side, you can see how Condenser fits into the protein quantification pipeline, where we input our MZML file or other files with MS1 and MS2 spectra. We run this through Condenser, we get our list of quantified analytes together with the spectra that belong to them, and finally we search these spectra with one or more search engines. So the interesting thing to note here is that what comes out of Condenser is already an order of magnitude smaller than what we put in, but at the same time we haven't thrown away any data yet. So this comes down to the central approach of our Condenser method, which is an approach we call Quantification First. And in order to understand why this is such an attractive approach, we first have to consider what the state of the art is. So the state of the art we call Identification First, where we start by detecting our MS1 features in our different samples. We assign the MS2 spectra to these MS1 features and then search these with some kind of search engine. And finally, we summarize this set of identified MS2 spectra into a matrix of peptides quantified across the different runs. So as you can see from this diagram, we tend to throw out a large proportion of our data before even getting to quantification. So typically this is more than 70% of the data, a large part of which are MS1 features to which simply no MS2 spectrum was assigned. And of course there's always the part where we have MS2 spectra that simply cannot get identified. So we saw an opportunity to change this around and came up with a concept that is known as Quantification First, where we, instead of trying to identify or put in peptide identification to our MS1 features, we first cluster our MS1 features into so-called MS1 feature groups, and we then also cluster our MS2 spectra such that we end up with consensus spectra. So the nice thing about this is that this allows us to do the identification step first after the quantification step. And this means that we first of all have much fewer spectra to search. And second of all, also the part that we cannot identify or that didn't get MS2 spectra assigned to them have been condensed into a much smaller set, which are much easier to query. Also interesting is an approach that has become more and more common, which is to subsequently search our unidentified spectrum by a second search engine, for example, an open modification search, and thereby increase the number of identifications we get. So the nice thing about this is that we can keep using different identification techniques without having to redo the quantification, which in essence also doesn't actually change. So I think most people agree that this is a very attractive approach, but of course there are several technical challenges that need to be met. So one of the first challenges is that we need to somehow align our MS1 features across the different samples. And normally this is done by using the peptide identifications to serve as nodes in our retention time alignment. However, in reality we don't need the peptide identifications to do this, and instead we can use MS2 clustering to figure out which MS1 features actually belong to the same analyte across different samples. And after having done this, we can actually align our retention time, and the nice thing about Condenser is that we also automatically can assign retention time windows and retention time tolerances based upon these alignments of MS2 spectra coming from the same analyte in different samples. And now we can do what is known as match between runs, where we assign, in our case, not actual peptides, but rather sort of MS1 feature groups across different samples based on retention time and precursor mass. So one thing that has been noted in the literature is that this process of match between runs doesn't come without its dangers. And one thing we therefore set out to do was to not only do match between runs as is, but also compute the probability that each of the matches corrects between different runs or between different samples. And the way we do this is by using so-called decoy features. And the interesting thing is that we don't just apply some kind of FDR threshold, but we actually keep the probabilities as they are. And this, of course, makes a lot of sense because some matches are much more likely to be correct, since we can see that, for example, the retention time barely differs, whereas other matches show a much larger retention time difference, and we might not want to trust these. So as I said, we don't set any thresholds, but instead we propagate the uncertainties using a Bayesian model. And for this, we use a package we have developed previously called Trickler. And here we input not only the peptide quantifications, but also, for example, the confidence we have that the match was correct between different runs, or that the PSM was actually correct. So what we can do now is to propagate these uncertainties all the way from feature level up to protein level. And in the end, we end up with a distribution over the fold change, which we can then use to assign significance or significant differential abundance or not. So this is all nice, but of course we want to see if this idea actually works in practice. And for this, we took a look at an engineered dataset of UPS spiked into a yeast background. So as mentioned before, we get our consensus spectra. We search this with a regular database search engine, in this case TIDES, which is a variant of Sequest. We filter out the unidentified spectra and then run an open modification search on this, in this case mod A. And as you can see, the runtime is much longer than for TIDES search, but on the right hand side we can see that there are actually significant gains in the identification rate, where the dark green bars represent the results from TIDES, and then the lighter green ones represent what we have added by using mod A as a second search. So this actually resulted in 56% extra peptide identifications, but in this case most of these modifications were most likely sample handling artifacts, so biologically not very interesting, but still a nice proof of concept. So as you might have noticed from the previous slide, the larger the cluster got, the higher the identification rates were. So this led us to investigate the largest cluster using the Novo search engine, in this case Novor. And in fact, we could find two proteins that were previously not searched for in our FAST-A file. One of them was a capsid of a known yeast virus, and second of all, there was a protease that wasn't actually listed in the sample description, but we could find this anyway. And then a second approach would be to use the abundance pattern. in this case, we compared the abundance pattern of our unknown analytes or unidentified feature groups. So we looked if the abundance pattern was the same as the spike-in proteins of UPS. And in fact, we could find a lot of modified UPS peptides. And often these came from quite chimeric spectra, which shows that the open modification search maybe is not as sensitive to detect these peptides in chimeric spectra as the regular database search was. And interestingly, we also found a big group of polyethylene glycols, most likely a contaminant, used in the UPS spike-in, but not in the yeast background, or at least not to the same degree. In the end, we mostly still care, of course, about the protein identification and protein quantification. And here we compared four approaches, 2 of them quantification first and 2 of them identification first. And we tested this on 3 different datasets, so the UPS spike-in dataset I referred to previously, but also 2 clinical datasets on the right-hand side. So we can see on a feature group level that we already frequently get a lot more identifications using the quantification first approach. And this also translates to more proteins quantified for the quantification first approaches. So as bioinformaticians, we also care a lot about the quantitative accuracy, not just as bioinformaticians, but also as experimentalists, of course. So what we did was we calculated the observed false discovery rate, meaning the number of false positives over the number of total positives. And we could confirm that on our UPS yeast sample, we could control the FDR below the reported FDR of 5%, and to do this at a very high sensitivity as well. Whereas MoxQuant combined with Perseus had a lesser sensitivity and also had relatively large problems controlling the FDR below 5%. So we saw similar trends when we investigated our clinical datasets. And here we compare 1 quantification first method in blue with 2 identification first approaches. And we could see that Condenser combined with our Bayesian protein quantification with Trickler could find much more differentially abundant proteins than both identification first approaches. And specifically in these 2 data sets, in the original analysis, the authors couldn't find any differentially abundant proteins at FDR thresholds. So they had to resort to p-value cutoffs of 0.05. And this is, of course, much less reliable than having these multiple testing corrected values. And also very compellingly, these differentially abundant proteins actually enriched for several functional annotation terms that seem to make at least some sense biologically, although further investigation is definitely needed. So to summarize, we introduced here our quantification first approach, which together with clustering allows easy researching and also deeper investigation of the data. We also saw that Condenser together with Trickler had a very high sensitivity, and this allowed us to extract information using actual FDR control thresholds rather than using some kind of p-value cutoff, which isn't as reliable. And something that I haven't touched upon yet, but what is also important to note is that Condenser is about 1.5 to 2 times faster than MoxQuant. Of course, you still have to do the search engine, although since we reduced the number of spectra to be searched by 1 order of magnitude, the combined time is still much faster than using MoxQuant. So we have already had a user report that he could process over 200 files, and we are currently working on supporting more than 1,000 files to accommodate the larger and larger cohort studies that are being executed at the moment. So for the source code and installers for the different operating systems, you can go to our GitHub page, where we also have some examples, both to install Condenser, to run it, and also how to combine this later with Trickler. So with that, I want to thank my colleagues of the Shell Lab at KTH, and in particular my supervisor, Lucas Shell. And last but not least, I want to thank all of you for your attention.